Krebs cycle is the name of the process which uses acetate derived from carbohydrates, proteins, and fats, and converts them into energy through a chemical reaction, oxidation specifically, in the form of ATP.
Several of the components were established by Albert St. Georgi in 1930, for which he received a Nobel Prize. The critic acid was finally identified by Hans Adolf Krebs in 1937, which received a Nobel Prize too.
The Krebs cycle has a lot of steps. First, the pyruvate molecules made of 3 carbon from the glycolysis are converted into another type of molecule called acetyl-CoA in a process known as pyruvic oxidation. This conversion occurs when the pyruvate is broken down by an enzyme, releasing a carbon atom which goes onto a form of carbon dioxide known as CO2. The 2 remaining carbon molecules bond with a coenzyme forming acetyl-CoA. Then, the acetyl-CoA enters the Krebs cycle. It initially combined with a 4 carbon molecule called oxaloacetic acid, forming a 6 carbon molecule of citric acid. Next, this acid undergoes what is known as oxidative carboxylation, which simply means that a carbon and a hydrogen are given off. The result of this 5 carbon molecule called alpha ketoglutarate. In the next reaction, this alpha ketoglutarate has yet another carbon molecule removed and is then transferred to a CoA molecule by the enzyme. After this process, NADH is formed. Then this molecule undergoes the removal of the CoA carrier, resulting in the production of succinate. Next, this succinate is dehydrated by the enzyme. The resulting product is formate. The formate is then dehydrated by the enzyme formase to form malate. Lastly, the malate is dehydrated by the enzyme malate, forming the original molecule from this reaction. NADH and NADHP are also produced. Once the molecule has been regenerated, the Krebs cycle can repeat. With the completion of this cycle, the electron transport chain (ETC) and the subsequent oxidative phosphorylation occurs, resulting in the production of 38 ATP, this providing the cell with energy.
The Krebs cycle evolved from pathways from amino acid biosynthesis. The design of this metabolic pathway, as it occurs in living cells, is the best chemical solution. It has the least possible number of steps, and it also has the greatest ATP yielding. Components of the Krebs cycle were derived from anaerobic bacteria, and it may have evolved more than once.
While this cycle is highly conserved, there is a significant variability in the enzymes between eukaryotics and prokaryotics. The conservation of some substances catalyzed in eukaryotes by the NAD, while prokaryotes employ NADP. Other example of this is some prokaryotes use the quantitative-dependent enzyme and eukaryotes use the NAD.